So we've been really zooming into vascular tissue so far, and we've looked at how water moves through the plant body, how it gets to the roots from in the soil, then how it moves through the plant body and out as water vapor through the stomata. We've looked at phloem translocation, how does sugar get moved around in the plant. We're gonna keep going with vascular tissue and root tissue in particular, because now we need to think about mineral acquisition. How do they get their minerals? And that, again, we're diving down into the soil and looking at what's happening with the root and the nearby soil. This little blurb here was written back in quite a while ago, where they are talking about depleted soils. Most of us today are suffering from certain dangerous diet deficiencies, which cannot be remedied until the depleted soils from which our food comes are brought into proper mineral balance. So this person, who is from the 74th Congress, a Senate document saying that our soils are poor in mineral nutrition. We need to get this fixed. The food that you eat currently doesn't have enough nutrient density in terms of minerals, as well as other things. And so this was kind of the alarm call that was put out, legal document, and this was way back in 1936. So this is a little alarming that that was being raised back that long ago. And I would argue that we still have to think about this currently. And when you think about soil systems and how we grow our food, the conventional way of growing foods in large agriculture systems, a lot of the minerals have become difficult for the plants to get access to in more than one way. And if they have problems getting access to minerals, either because there's not enough there or they're locked up in a way that their roots can't really access them, then the plant suffers, its health will suffer, and then you eat that plant. It's probably not the best for your health either overall. You could do better, right? And so this idea of how is the plant getting access to minerals is a huge topic. It's a hot topic. This is not going away. People are very concerned about this. There's been a lot of even common literature out there, movies and books written about the idea. Kiss the Ground is probably one of the newer documentaries that's out with Woody Harrelson narrating it. Very good one. And I think it was on Netflix for a while. I don't know if it's there anymore, but very, very well done. And then these are some books that have been written. Dirt is another DVD documentary that's also very good. And so all of these resources are out just for, regular people to watch or read. And so it's getting out there that we need to kind of rethink how we grow our crops, especially, and that if we don't have a healthy soil, we're not going to have healthy plants. And then, of course, we eat those plants. So it just keeps going on up the line in terms of we can suffer from that too. I found this quote very interesting. Dr. Pauling, he's a Nobel Prize winner, and he is saying that mineral deficiency, you can track human ailments back to mineral deficiencies being a real part of that process, leading to that disease state. And so it is something to really think about and consider, especially if you're concerned about eating food that might not be as nutritious as you thought it was, and how to approach that to get better access to better food. But from the plant's perspective, they need minerals, and that is the soil system. So they're mining the soil system for those minerals. So we're going back down into the soil and thinking about roots and uptake of minerals. So as they're doing this, they are getting access to minerals by whatever is dissolved in the soil water. And that's what it has access to. So if there's minerals dissolved in the water system in the soil, great, the plant can take those up. As it's taking water up, it'll take up dissolved minerals. Great. And so they, remember, of course, we'll talk about how they take in CO2 and make sugars off of that later, but certainly they're getting their carbon needs met through photosynthesis, but they still need all those minerals. And so they can't make those, they have to get that from the soil system. And roots are incredibly dynamic, root systems. I think I mentioned already, maybe during the first week, that for many, many, many plants, what you see above ground is not as big as what you would see below ground. So some root systems, a lot of root systems are massive compared to what you see above ground. Kind of like that, the iceberg analogy, the tip of the iceberg idea, is a good analogy. And they're continually growing and dying, new roots being made. They forage, for lack of a better word. I think that's a really good way to describe it. They forage by growing. They look for food in that way by growing. And when they find a patch of good, yummy minerals, they'll slow down their growth and just kind of hang there and sop up the minerals. And then if that area starts to get devoid of minerals, they'll start to grow out again, searching for more nutrition. And so it's a very dynamic area. Lots of organisms in the soil love to hang out with roots because roots are what we call leaky. They just naturally leak out all kinds of organic, well, not so much inorganics, a lot of organics. They tend to leak out some inorganics, I'm sure. And that is very attractive for the microbes in the soil. They love that for their nutrition, all the nematode worms and other creepy crawlies. And so soil systems and root systems in the soil around it, very, very active. Lots of activity, lots of organisms. Root systems, they often show this comparison. Taproot on the left, where you have a big fat central root, and then you have lateral roots coming off of that, but you can clearly tell the difference between a taproot and the lateral or horizontal roots. The right picture is the fibrous root system where they all pretty much look the same. And then I just want to emphasize there's all kinds of gradations between these two extremes. It's not either or. You will get different designs depending on the plant species. Depth of root systems can vary tremendously as well. So some plants have very deep root systems, others don't. That's also heavily influenced by their environment. So how good is the soil? Is the soil only good to a foot? That's where most of the roots will be. If the soil is good for growth and getting access to nutrients and water deeper, then the roots will be deeper. Places like the tropical rainforest along the equatorial line, that soil system just geographically, it's not very thick, not very deep. And so pretty shallow root systems in that part of the world. And so it really does depend which plant you're thinking of, where are they growing, where are they adapted to, and that will certainly influence their overall root design. Here are also some other ideas to think about that some root systems we certainly take advantage of because they store up food really well. And we can get good nutrition from carrots, tap roots such as carrots or tuberous roots as well. So sweet potatoes and others. So this idea that you have a static root system, get that out of your head. It has to be thought of as a very dynamic system, heavily influenced by their environment and the soil system. And you can influence the roots of your plants, whether it be your lawn or potted plants that you have in your home, by how you water those plants. And so, for example, with lawn grasses, if you water them fairly infrequently, but when you do water, it's a very deep, nice soaking event where you let the sprinkler go for quite a while. That tends to create root systems that are pretty long because you soaked the soil system, and so the roots will start growing down and kind of tracking that water. If you decide to water your lawn like every other day, but just for like 5 minutes, and you're kind of stingy on that, then the water doesn't go down very far and the root systems won't either. And so how you water your plants can actually pretty nicely influence the root system. So give that a thought, whether it be a potted plant or not, you can influence that. So here they're showing the difference between an irrigated soil, and that's like what I was saying, a nice soaking irrigation where you really give it a thorough watering. You can really manipulate the idea of the shape and morphology and the depth of the root system. And again, trying to impress on you different root system, different plants, whether or not they're watered well or not, you can get a pretty profound influence on their morphology. So roots are very important. Certainly, they're taking up water and minerals. They anchor in the plant into the soil system to keep them anchored in and stable. They're used for storage. And it's really they're trying to show here by the arrows of where they're zooming in, they're trying to show that the root hairs are very, very good. They're kind of like the workhorse of the root system in terms of getting water and mineral uptake. So this is a longitudinal slice of a root system, and they're trying to show the different zones. And I would like you to know the different zones. And so when you look at this, you have the, well, this gray area here that they have, that is the mucilage, mucigel they call it. And it is a nice slimy covering to the root, which makes sense when you think about it because soil is so abrasive that as the roots are growing through the soil, you can imagine if they don't have some kind of slimy coverage, it would really cause a lot of damage to the root cells. And so having that makes a lot of sense. And then you have your apical meristem region. So the meristematic zone, that's where you have those precious cells that are pretty actively dividing on a regular basis, and you want those well protected. So that's another reason why you have a root cap over that. So at the very end, you have a root cap covering the apical meristem region for protection. And then on that, you also have the mucilage covering it. And then behind that, you have the elongation zone. Okay, the elongation zone, that's literally where you see cells starting to grow bigger and starting to form their mature state. And then a little bit further back is where you get your so-called maturation zone. That's the cells that are fully developed. They've been morphed into their final role. So some of it will be just parenchyma cells, some will be vascular tissue, some will be root hairs, epidermal cells. And so that's kind of the, and when you look at a root tip, this is all crunched down. This is very much kind of blown up. But when you look at a root, it's really a very small area of near the root tip that we're showing this kind of idea. So most of the root is mature roots, cells, and they're actively doing their jobs, respective jobs. All right, so you have all this root buried in the soil. So if you haven't taken a soils class yet, you know, well, if you have taken one, you know that soil is very diverse in composition, mineral availability. And when I took a soils class, I took my first soils class actually at the grad level, and it was up in the forestry department at Michigan Tech University. And I thought, really, a whole semester on soil? And I thought, this is going to be kind of boring. And actually, it was one of my favorite classes. It was such an eye-opening class in terms of what the different soil systems are like. And yeah, it was amazing. So when you take that class, I hopefully you'll say the same thing. It's not at all. I learned so much in that class. A lot of misconceptions were put to rest. So here you have to think about inorganics. That's what we're zooming in on. We're thinking about all the minerals that are in the soil. So these inorganic particles, calcium, magnesium, potassium, and then certainly there's organic particles that are there from dead and decaying organisms, other plants, so on and so forth. You also have liquid, hopefully. You have lovely water, rain events going on, so you have water in the system. And in that water, you'll have dissolved minerals. And then gases too, don't forget. Whatever we have above ground, it's below ground too. plus there's also other gases that tend to get captured in the soil system and slowly percolate through up into the atmosphere. Besides oxygen, carbon dioxide, and dinitrogen, you have hydrogen sulfide and methane and nitric oxide, nitrous oxide. There's all kinds of other, this is just a partial list. But when we think about the idea here of the soil system, it's the actual soil particles that serve as a reservoir for the minerals. So these minerals are actually attached to the surface of the soil particles, and they can be attached through bonding to the soil through these, primarily because the soil, when you look at a soil particle, it's negatively charged. It has a definite negative charge. Doesn't really matter what soil particle you look at. And it has to do with some of the chemical groups that are on the surface, but it is a negatively charged soil particle in pretty much any soil system. So there's carbs that give it a negative charge. There's proteins that give it a negative charge, carboxyl groups, phenols. Okay, so it's a negative charge. The other thing about soil particles that you have to think about is the size of the soil particle will influence surface area to volume ratios. So the smaller the particle, the greater the surface area to volume ratio. Big particles like sand have a smaller surface area to volume ratio. And so our surface area, we have to think about that a little bit, because that's where the particles are. They're on the surface, are the minerals, or they're on the surface. So here they're trying to show some soil particles. They're saying these are clay particles, so those kind of square things. And what they're trying to show is that you have the minerals or the different colored balls that are attached to the surface of these clay particles. And then you have a plant root over here. So I think this is a good graphic because it's really driving home the point that the only, well, plant roots really can't get access easily or not at all to minerals that are still attached to soil particles. It's not like the root can just grab that off there and say, I want that. All right. So if the minerals are attached to soil particles, plant roots cannot really access that. There's a couple of exceptions, but in general, they have a really, really tough time with that. So what the plant has to wait for is for these minerals to detach from the surface of the soil particles and get into the water of the soil. Once that happens, they'll dissolve into the soil water, and then the roots can take it up. As they're drawing water into their body, the minerals that are dissolved come right with it. But the trick is now, how does that happen? How do we get the soil particles, the minerals to come off the soil particles? And that there's a couple of options to consider. And basically there's two big ideas. There's a process called cation exchange, which I'm sure you'll learn more about in soils class. And then also microbes. Microbes are helping this process pretty greatly. So in a healthy soil, healthy for plant growth, there are plenty of microbes hanging out with the plant roots. And so plants are hanging out with all kinds of microbes. Many, many, many of them are very beneficial to the plant. They're on the surface of the plant, they're inside the plant, just like we have microbes hanging out with us. All right, it's very, very analogous. And certainly there's all kinds of microbes in a healthy soil. There's all kinds of microbes in the soil system as well. And so it's dubbed the plant microbiome, just like we have a human microbiome. And these images that you're looking at here on this slide, the Petri plate off to the right here, you can see that there's all kinds of different looking bacterial and fungal colonies growing on this agar plate. So this was a soil sample that I took and diluted and then plated out onto an agar, spread it around and let it incubate. And so all these different colors, different looking colonies, those are all different species of microbes. So you can see there's a lot there. It's chock full, very beautiful mixture of microbes. And then this lower picture here, what this is actually showing is a leaf that's been placed on a tissue culture medium. And you can see this fungal hyphae growing out of the leaf tissue. So these are fungi that normally live inside the leaf. But in this artificial situation where you've cut the leaf and put it onto this medium, they start to grow out of the leaf tissue onto the tissue culture medium as they search for nutrition. And so these are endophytes, what we call endophytes. So when you think about what's inside the plant, what kinds of microbes are in there, we call those endophytes. And it can be bacteria, fungi. In this case, it's fungi that's being visualized. And so you'll do tissue culture next week, I think, in lab. You'll get to try your hand at that. Hopefully you won't have this situation, but there's no way to really eliminate the risk because they're inside the tissue of the plant. And the only way to kill them off the ways to do that would also kill the plant. And so it wouldn't work. And so it's always kind of a gamble when you are doing tissue culture that you might have endophytes starting to grow out of the tissue that you're working with. But we have a method down that's pretty good. So usually it's highly successful. So the root system, I like this analogy, root systems which absorb nutrients and are directly exposed to soil microbes are functionally analogous to the animal gut. So to our gastrointestinal tract. So they're saying that it's pretty equal in terms of their functions and how they interact with microbes. I thought that was a very nice way to think about it. And so this is a blurb in this microbe magazine where they're looking at studying the plant microbiome because they really feel just like with the human microbiome. If we can better understand it, we can improve health of the host, whether it be human or plant. And so it's a big push right now in research, looking at the plant microbiome. So microbes are hanging out with the plants, whether they're inside the plant, on the plant, right near the plant, right near the root. And the ones that are in the soil are actively helping getting those minerals off the soil particles and into solution. They're doing it for themselves too. I mean, they need minerals too. So it's actions of their own doing for their own nutrition, but then of course some of it gets brought into the plant. So microbes are hugely important in that process. Cation exchange is another process that's just kind of chemically going on, and it just kind of constantly goes on. So if you think about minerals absorbed and attached to the soil particles, so we said soil particles are negatively charged. So what kinds of ions are going to bond to the negative charges? Cations. So that's what we're really zooming into. We're thinking about cations. Now, there are minerals that are negatively charged that the plants need, for sure. But they can't attach to the soil particles. So that's a problem. And we'll talk about that in a little bit here. But when we talk about cation exchange, what we're thinking about is a cation is attached to the soil particle, and now another cation is going to come along and exchange, like a one-to-one exchange, and bump. I like to think of it as being bumped off the soil particle, and that other cation takes its place. So cation exchange, we're talking about bumping one of the cations off the soil particle, another one taking the place. So this can be done. They know like the binding affinity of all these different cations, and this is called a lyotropic series. There's lots of them you can look up. This isn't just one example. But in general, the way to read lyotropic series when they're printed out like this is that The binding affinity goes from high on the left end to lower and lower and lower as you work your way right. And so what this is saying is aluminum in this series, aluminum has the highest binding affinity for the soil particle. That means it's going to be hard to bump off. It's really nicely bonded to the soil particle. Very tight and high binding affinity. On the other hand, sodium is very easy to bump off. It has a low binding affinity. So if calcium comes along and sodium is there, calcium can just say, hey, get off, it's my turn. And so sodium will get bumped off, calcium takes its place. So the way to interpret this is anything to the left, so to speak. So aluminum can bump off all of the ones listed to the right. The proton H plus can bump off everything to its right. So that's how you interpret this. Okay, calcium can bump off everything to its right. And so you have this process going on. If you imagine water, a rain event, so water is percolating through the soil, nice soaking, lovely rain. And you have dissolved minerals, flowing in that water. They're there, they're dissolved, and then they're flowing by the soil particles. You can imagine where you're going to get just cation exchange happening. As long as there's water in the soil system, dissolved minerals, you can have this exchange of minerals going on. So here they're trying to show a clay particle and all the negative charges on it. And then they're showing, in this particular case, they're really kind of zooming in on the proton. So they're saying that, so this bottom reaction, by the way, what they're implying here is that, the roots are busy going through respiration, just like you're respiring right now. You're doing your thing, getting ATP made in your cells, and then you give off CO2. So plants are doing similar things. So they give off CO2 that goes into the soil, reacts with the soil water, and in that process, you get bicarbonate and a proton as a product. So then the H plus, the proton, can now potentially bump off. Here they're showing potassium getting bumped off and the proton taking its place. And then if you finish the thought, it's like, okay, so the proton bumped off the potassium, and now they're showing the potassium getting taken up by the root. So this cation exchange allows for these minerals to get bumped off the soil particle into the water, and then the plant can take it up. Make sense? So soil particles and their surfaces are important for the root systems to be able to do uptake in this cation exchange going on. You got microbes there doing their thing. And so the size of those soil particles can vary. So here they're showing just 3 different types of clay, the morillonite, illite, and kaolinite. And you can see that their size varies a fair bit. And then the last row, if you look at, the first row where you're looking at size differences, and so the morillonite that has the smallest size, right, has great cation exchange capacity. So if you garden at all or growing plants and you take a soil sample and send it off for analysis, they usually spit back the data of your cation exchange capacity. And you should pay attention to that. You want to see where it falls. And in general, a high cation exchange capacity, that's what you're going for. You want that because that allows for nice exchange going on, pretty constant. And that means that the particles, the minerals getting bumped off the particles, that's happening quite a bit. Then those minerals go into the water, and then the plant takes it up. So it's good for the plant overall to have high cation exchange capacity. Here they're showing a soil particle, that brown blob in the top, very top is your soil particle. And they're saying there's some stuff attached. They're just putting X plus, so they're being very vague. But what they're trying to show here is that depending on what's going on inside the root cells, you might have to expend energy to allow for mineral uptake. So here they're showing ATP being used in order to push protons across the membrane. And if the plant does that enough times, you're going to get a proton gradient across that membrane where you get more protons outside the root, less in. And that is going to help with cation exchange. Plus, here they're also showing it's also good for symport, where you have a proton coming through transport protein, and it allows for the intake of a mineral, X plus, whatever that is. So they're trying to imply here that there's some nuance to this. I'm not getting into huge detail, but sometimes the plant does need to expend energy. Not always, but sometimes. And that can help with cation exchange because it creates that proton, which can help bump a mineral off. And then that proton gradient can help with uptake of the mineral because of the symport process. So that's what this lower image is also showing. It's pretty much the same thing. So that's kind of a little nuance to this whole process. So cations, great. We have this, cation exchange going on. Microbes are there helping with that. But there's several nutrients that are negatively charged that the plant also needs. But if the soil particle is negatively charged, it's like, how's that going to work? How can there be a reservoir? Basically, there isn't. So there's no soil reservoir for negatively charged minerals or nutrients. And that is one of the huge reasons why farmers apply nitrogen in such large doses, because many times it's in the form of nitrate, NO3 minus. And that is very temporary in the soil where the roots are. So if you apply it onto your field, it gets watered in. It starts percolating with the water through the soil system. If the roots don't take it all up, it just keeps going right past the roots. It doesn't attach to anything, and eventually gets to the water table, can cause some problems there. So if you get too much nitrogen in your water table or getting dumped into nearby streams and lakes, then you get that terrible eutrophication going on, which you've learned about in ecology classes, where you get too many nutrients in this case, in a lake or in the water table. And that causes all kinds of issues, kind of a cascading suite of issues, including a lot of fish end up dying off and other organisms that need high oxygen levels. It's a real cascade of issues. So definitely not a good thing. And also for the grower, if they overdo the nitrogen input, that's a cost. They are losing out money for that. So that's not good either. And so knowing what your nitrogen is like in your soil system is pretty important. And then being able to add in nitrogen in the right way and with the right amounts is very important. So again, just trying to show that you have all these positively charged things able to attach to the soil particle. But in figure B and C here, you see like NO3 minus, nitrate, you see sulfate compounds have negative charge, chloride ion has a negative charge. So those just never attach. They're just staying in the water. and can percolate down past the root system, and then the plant won't have access to it anymore, but it goes out into the environment. So that's extra issue to think about. Okay. Another thing to think about is that soil pH, it can vary, but in general, they've sampled lots of different soil systems all over the world, and the general consensus is that most soils are slightly acidic, especially soils where we grow food, crop production. Those types of soil systems are definitely slightly acidic. So remember, pH is dependent on the concentration of the proton, H plus. So it's the negative log of the proton concentration. That's how we determine pH. So H plus plays a heavy role in this. Slightly acidic soil, pH 5.5 to 6.5. So that's what the roots are dealing with. They're dealing with a slightly acidic. I would like you to memorize that range, please. Okay, 5.5 to 6.5. That's kind of the range that they give for most soil systems. And so that seems to be the range, the pH range, where most minerals are most available to the plant. And when we say available, what we're saying is that there's plenty of microbial action and cation exchange going on where those minerals are getting put into the water of the soil and they're more soluble. Okay. So that's what you see that term a lot, that these minerals are available. So what you have to think about is, oh, they must be dissolved in the water in the soil. That's what's going on. The other thing with slightly acidic soils is that fungi are more prevalent. Fungi like acidic conditions. I've worked with some fungi where they love like pH 3, pH 2, which is a little bit more extreme than this, but fungi are happy in acidic conditions. Now that can be a good thing for the plant and is a good thing for the plant in many, many cases, but it can also be a bad thing. Because when you ask a plant pathologist who studies plant diseases, what's the number one group of organisms that cause the most plant diseases? Fungi. Unfortunately, fungi. So fungal pathogens have an advantage in slightly acidic soil. It's their comfort zone. They're good with that. But there's all kinds of beneficial fungi that also like slightly acidic conditions. So it's kind of a double-edged sword. This figure is trying to show you the availability of minerals under different pH. And so if you go pH 5.5, whoops, a little off there, 5.5 to 6.5. So if you look at that area on the x-axis and then just draw your eye up, you can see that most of these lines are pretty stout. They're thick, whereas some of them thin out as you go into higher pHs. So manganese, for example, is much thinner at pH 7.5. That means it's less available, less soluble. So wherever you see a thick line, that means it's nicely available, which means it's easy to have it into solution, into the water solution of the soil system. So there's ways to manipulate that to some degree. This is the same image, it's just colored. So if you like to see that one instead, that's the same idea. And so when they did all these samplings of all these different soil systems and they're like, yep, it's slightly acidic. It seems pretty much consistent. And, there's some exceptions, but pretty consistent. And so if you think about it, well, soil systems get rained on, hopefully fairly frequently, enough to keep going for the plants. When that's happening, water is mixing with the CO2 that we just talked about a few slides back. So when you get that happening, you do end up with products like bicarbonate and then the proton. And that's what's influencing your pH. So the more protons you have in the soil system, that's driving your pH more acidic. And so it seems like there's consistently enough of those protons that you get this 5.5 to 6.5 idea. As the soil is acidic, you also have these compounds being capable of forming under slightly acidic conditions. So aluminum can actually get off of the soil particle and start reacting with hydroxyl groups, and iron as well. And then as that's floating around in the water system, you can have it reacting with phosphates. And then you get complexes like this that are insoluble. Now, it's not such a bad idea that aluminum becomes insoluble, which means if something is insoluble in the soil system, that means it's precipitated out of the water, which means the plant roots can't have access to it. By and large, it's not available to the roots. Well, that's okay for aluminum, because aluminum is toxic to plants, just like it's toxic to us if we get too much of that exposure. So that's fine. However, if it combines, reacts with phosphate, plants need phosphate. So now the plant has just lost its ability to get phosphate because of aluminum doing its thing. So it gets complex pretty quickly. If you take a soils class, I'm not sure how much they go into some of these reactions going on in the soil, but this is both good and bad. Having aluminum precipitated out, that's good. Having phosphate precipitated out, plants need that. And so that's a real problem. And it does create phosphorus deficiencies in the plant. So that's can commonly happen. You can manipulate that to some extent. So this is under like when this acidity is getting closer to like 5.5-ish. So if you want to raise it up a little bit and try to prevent this, you can add things to the soil. I've done that in my garden when I've found that it's getting a little too acidic. You can add calcium or magnesium, lime, calcium carbonate. All of those would help raise the pH a little bit. And growers do this on a massive scale too. It's not just for your home garden. Another, well, if you're curious, so for example, if you take calcium carbonate and add that into the soil system, what it's doing chemically is it's starting to react with the protons. So if you get the protons into, it's no longer a proton, it's reacted with the carbonate. Now you've removed free protons from the soil system. So that's what's allowing your pH to come up and not be so acidic. Get rid of the protons. That's the goal essentially, and that will help raise the pH. Okay, so there's a lot of dynamic going on chemically in the soil. And that's for a soils class to get into a little bit, but I did want you to be aware of the cation exchange idea. And the other big idea that often does not get talked about is that microbes are there doing their thing. And they're hugely important in this process for the plant to get access, for minerals to get solubilized, and then the plant being able to take it up. And agricultural systems that have been intensively farmed in the more conventional way, where they use inorganic fertilizers, it often creates a situation over time where it causes buildup of toxic compounds that kill off the microbes. And they're actually starting to refer to some agricultural land as microbial deserts, because there's so few microbes compared to what they used to be like, that it's essentially a desert. It's a wasteland. There's not enough. And so putting plants into those soils, it's going to be really hard for them to grow well and get a good yield because the microbes aren't there. So it's not just the cation exchange capacity and the cation exchange process to think about. Microbes play a huge role in this, and it needs to get talked about a lot more than I think it's currently being talked about in like soils classes and in agricultural teachings. So keep that in mind. The microbes hugely matter. The next topic we're getting into in this slide set is microbes. We're going to talk about mycorrhizal fungi and how incredibly important they are to plants. But that's a kind of a big topic, so I'll stop there. And we'll get into that on Monday. So have a good weekend. It's homecoming weekend. So be safe with all that, but have fun.